Yeah, welcome to this Sam Payne seminar, and it's a great pleasure to introduce Sam. So I've known Sam for quite a few years. He got his PhD from UCSD in computer science, but with a focus on biology. And then he went to the Craig Venter Institute for 2 years and worked on combining genomics and proteomics. And after that, he went to Pacific Northwest Laboratories, where he's been for a few years. And there he is heading up the bioinformatics and focusing on and being part of a bigger group that does a lot of cutting-edge mass spec instrument development, and they're also involved in several larger biological projects that generates large data sets, both from human samples, but also from a lot of bacteria.
So it's a great pleasure for me to be here in New York. I used to live in Washington, D.C., as he mentioned, at the Venter Institute, where they kind of pioneered all the genome sequencing. And Craig Venter is the name for that, and his company was an awesome rocket to be on. And then Pacific Northwest National Labs, my boss there, Dick Smith, made most of the mass spectrometers that are on the market today. So he is an analytical chemist, physical chemist, and his great love and desire is tinkering and building things. And so he'll spend years building something, and then you'll see it in a couple of years in the next line of commercial instruments because he's licensed it and he's moved on to the next thing. And for a long time, he did really great work, and then he started getting into new instruments that really needed an algorithm person. The instruments were new enough and different enough that he really needed a dedicated algorithm group. And that's where I came in, and I said to him, the problems that you're going to have with these new instruments is that you're going to release them and nobody's going to be able to do anything with them. It's kind of like the PacBio problem. They have their own in-house software group because what they're doing in super long reads is so different. They need people dedicated to that project. I thought I'd start off and show you a little bit about our group. This is, we call the Omics group. So anything you can measure that has mass, we measure. So we measure metabolites, proteins, lipids, anything that you can measure that has mass. That's what we do, which is why it's omics or panomics. It's not just proteomics, it's everything. And the group here is as big as an academic department, and it's run in a very team science way. I love team science, personally. I moved to PNNL for team science. I like that. This is, if I get a pointer here, this is me standing behind Dick Smith, who's the boss of this. But you have, in this group here, you have 20 or 30 people who are independent PIs. We have our own funding, we have our own projects, we do what we want to do, but we're part of the big group because we love working as a group. We are Team PNNL. We're not a degree granting institution, so we mostly thrive off of postdocs and internships and people coming to visit for a while. It's a fabulous place to be. Since no one has ever heard of Richland, Washington, I thought I'd show you. We are the non-Seattle part of Washington. We're in the middle of Washington wine country, if that's an attraction most people love. It's not the big green Pacific Northwest that you might think of, but it is rolling hills and apple orchards and wine everywhere you look. So I love it because it's a small town, and I'm from Kansas, and small towns are wonderful. My sister, who lived here in Manhattan for many years, loves New York. I got these small town genes, and so we're different people. I thought I'd talk a little bit today about big data and what is big data and what are the concerns that I see on the front line, and then my experiences with large clinical consortia, and then finally some solutions. This is where I wax quixotic in the solutions for everything. So big data. When you talk about big data, people usually talk about the 4 V's. These are the nerds who say volume and volume and volume. And then you get into velocity and veracity and variety and all of these things. This is how people fund grants. They make piles of data. And there's big pipelines, and we throw some math and chart foo on you, and it's a pile of data. I told David last night at dinner, our servers have more than we can actually publicly give away. That's not because other people don't want it, it's because they don't have 300 terabytes that they can host on a web server. We tried for many years to host 300 terabytes on a web server, and it is insane. It just doesn't work well. We have more data than we can give. And this is data. We make data. And the problem that I've ran into in the last several years is that discoveries are not made that way. Discoveries are made in a very calm, reflective, pensive moment where you sit and think. This fundamental dichotomy, this disconnect between piling data. You know, I've been to the Genome Center in St. Louis. I worked at JCVI where we have and made incredible amounts of data. They have buildings just to hold servers for their data, right? That compared to the clinician who you work with, who sits and wonders how this one pathway is giving the phenotype, right? This has been the frame for my last 6 months' obsession, is how do I solve this gap? What can I do to help these people? Because I can make lots of data. I'm good at that. But I'm waiting for the shoe to drop when people realize that piling up data doesn't actually cure things, right? You need some help in that specific arena. So that's why I'm going to talk a little bit about my personal experience on a project for ovarian cancer and then what I think real solutions are that we could draw to this. As a personal curiosity, who in here would self-identify as a computational person? Who in here would self-identify as a not computational person? All right, I love all of you. As David said, I have what amounts to a joint PhD. I have a computer science and biology degree. They called it computational biology or bioinformatics. And so I had to take the beginning biology graduate student courses, and I tested out of all the beginning computer science graduate student courses, but they required proficiency across all of it. And it's an incredible divide, but one I think we can do better on. Just in terms of a little bit of background, chromosomal abnormalities are things that we are or should be very familiar with. This is Down syndrome, where you have a third copy of chromosome 21. You have 3 of these. So in addition to the one you got from your mom or your dad, somebody else had a friend, and you get a third. That causes a variety of problems for Down syndrome specifically. There are other trisomy diseases, although they're more lethal. In general, trisomy is a very hard thing to have and survive, actually past the blastula stage. So this is a cancer karyotype. This person is close to tetraploid, although they have only 3 copies of chromosome 2 and 5 copies of chromosome 13. Cancer in general is large-scale problems. You have many, many chromosomal abnormalities. Here we're looking at big karyotype issues. I'll point out that you only have 2 copies of chromosome 6, but part of chromosome 6 is over here attached to chromosome 4. So when you think about it at this big scale, this is one of the issues you see with cancer. Another issue you see with cancer is called chromothripsis, where your chromosomes have been split into many, many, many thousands of pieces. You don't have anything big anymore. You just have small pieces everywhere. Looking at a much finer grained detail, we talk about things called a copy number aberration or a copy number alteration or a copy number variation, whatever your word soup of the day is. Basically, if the reference genome has 4 genes, you can have 2 copies of chromosome of gene B. Or you could have 3 copies of gene B. Or you could have this gene CD repeating, or you could lose gene B. Right? There's all sorts of small-scale problems that could happen in your chromosome. And the generic word for these small-scale problems is a copy number aberration, or what I'll call in the talk today a CNA. Each of these comes with some amount of dosage problem. You have too much or too little DNA copies, and that is reflected in having too much transcription, too much translation. You have essentially too many proteins for this locus at the end of the day, or too few. CPTAC is the large consortium that I work in. This is the Clinical Proteomics Consortium run by the National Cancer Institute. It is associated with TCGA, which I think many people are familiar with. And our goal is to proteomically characterize tumor cohorts. So I work on an ovarian cancer project. David here and Kelly work on a breast cancer project. And in each of these things, we got tumors from the TCGA, and we will take those and do proteomic analysis on them and derive new knowledge above and beyond the genomic information. So what I'm showing here in this one is the publication that's out first from the colon group at Vanderbilt, and they found a new subtype of colon cancer. It wasn't available, didn't see it at all in the genomic data, but they found this new subtype which correlates with hypermutations and a severe p53 mutation, and also a loss of chromosome 18q, the entire arm. So although those things showed up a little bit in the DNA data, they were not strong enough to be declared a separate subtype. And so the proteomics data adds to this, and we can say this is a new subtype. It's driven by a certain set of genes, and it is targeted for therapy by another set. And at the Vanderbilt Cancer Institute, that's what they're tackling down. It's great work, and it comes from insight gained on proteomic data that was additional to what had already been seen by TCGA. So I work, as I said, on ovarian cancer. Ovarian cancer is a disease that's very severe in terms of mortality. The PI of this project in our group, Karen Rodland, describes it quite graphically in terms of having kilograms of tumor inside of you. So it's a very debilitating thing that we want to address, and the patient survival is about 50%. So TCGA has already published 2 or 3 years ago an ovarian cancer paper where they looked at genome or transcriptome or methylation data. And one of the first things they found was there is a large-scale snafu with the chromosome, but not anything specific or focused. So in this figure, what I'm comparing here is this is glioblastoma. This is from their Nature paper. And in glioblastoma, you have an amplification of the entirety of chromosome 7 and a deletion of the entirety of chromosome 10. That defines glioblastoma. It's in almost every patient. You can see pretty easily a concentration. And the rest of the chromosomes, normal-ish. Ovarian cancer, red and green everywhere. Everyone has a different set of problems. So they didn't have any simple solution for ovarian cancer. They just said there's large-scale, widespread problem, and we can't focus on it. The Nature paper there was actually more descriptive than solution-oriented. So what we're doing is looking at the effects of copy number changes, which you saw on the last slide were widespread, on protein abundance. This is work by my postdoc, Debjit Ray. And the general outline is that we're going to correlate copy number changes with changes in protein abundance. We're going to look for functional enrichment of proteins that have been changed and implicate those for some sort of driver mutation. All right, so I got to point out this one because it's, I'll come to the other side just to make you guys not feel lonely. Each dot represents a single person or tumor. And on one axis, you have their copy number for a specific genome locus. So the CALU locus, this person has relatively less of that DNA piece. And then on this axis, I'm showing you the protein abundance for that same person, for that same protein. So the positive correlation you hear is exactly what you would expect. You have more of that locus in DNA, you end up having more of that protein in your cells. How that affects other proteins is actually the really exciting topic, because if you have more of one protein, proteins don't act in a vacuum, they interact, and so it is going to be causing problems with its interacting partners. I like to use the example of the NF-kappa B pathway where you have an inhibitor, I-kappa B. If all of a sudden you had more inhibitor, NF-kappa B would be less able to do its job of transcriptional regulation. If all of a sudden you had less inhibitor, NF-kappa B would be going wild in the nucleus. And so we want to talk not about what is in genetics a simple cis effect, a locus and its own protein, but we want to talk about a trans effect. Trans effect is any locus and any other protein. So here, just to show you for fun, I have the TGIF locus and other proteins. Sometimes you see a positive correlation, sometimes you see a negative correlation. This would be played out in biology about whether this is a positive regulator or a negative regulator. whether we work to promote each other or inhibit each other. Because we're bioinformatics people, we can do this ad nauseum to everything. We can just throw correlations on the chart, and we do. So we do all copy number locuses compared in a correlation to all protein abundances. And then we filter these by some p-value that's corrected. And what you're seeing in red dots is whether they have a positive correlation, a locus and a protein. In blue dots is whether they have a negative correlation, a locus and a protein. The big strong diagonal there is the cis effect. My locus affects my protein. Anything off diagonal is a trans effect. My locus affects someone else. The most striking thing to me, what I immediately grabbed hold of, are these big vertical stripes. The big vertical stripe here means that as a locus, I impact lots of other proteins. My change causes a pervasive change for other things. So I've blown up a little bit of this, and you can see, let's look at this distal end of chromosome 1. There is a very white background here. Very few dots of effect. So those locuses, they do change themselves. Down here, this part of the diagonal. They don't really change anyone else. You look on chromosome 2, there is a big and a strong effect of hundreds, actually 300 or 400 other proteins who are affected by changes at this one locus. And that's what we want to focus on, because these are the potential drivers. Changing this locus changes the landscape of biology in the cell. So what I've done here is I've simply summed, and you'll see the numbers of affected proteins. In green on the top is anything that is not on the same chromosome, which is how I call trans. And red underneath is anything on the same chromosome. That's just an easy way for me to look at cis versus trans effects. And there are some prominent spikes. So this one on chromosome 2 that I showed you, next door on chromosome 1, remember there's almost nothing happening there in trans. Also, big plots on several other chromosomes. We want to kind of explore these and ask, are these things that functionally lead to ovarian cancer, or that can be functionally related to ovarian cancer? One of the great things that I like about this graph is the places that are really impacting, as by this graph, are not actually that common in ovarian cancer. So the piece I showed you at the very beginning that showed changes everywhere, it didn't show very many common changes. The only real common change that we could see was chromosome 16. That's one they bring out in the paper. And even though chromosome 16 might have a problem, not the same problem, but a problem on 50% of the ovarian cancer patients, it doesn't affect many other things. It's a relatively benign change in terms of the molecular biology. Not many other things get impacted. But chromosome 2, these abnormalities affect maybe 20 or 30% of the population. They really strongly affect the biology of what is happening. So that, for me, is a different definition of a driver, but allows us to think about biology instead of numbers. We're doing that as we speak, yes. So this analysis has been done on the colon cancer paper and was not this drastic. And so in order for a control, what we're going to do is we're going to take the HapMap data. So Mike Snyder at Stanford took 95 individuals who were part of HapMap, who, as being part of HapMap, have a whole ton of work up on the genome, and he did proteomics on them. And he published that last year, where he looked at QTLs that are cis only. So we want to expand that and look at trans. And the other thing is, cancer is a disease of dysregulation. It is a disease of problems. And so I am allowing the opportunity for this to present some sort of problem that isn't typical. We say, I would not have expected that this would have such a dramatic effect, because that's what cancer is. One of the things that I thought people would ask originally is, what is the difference between protein and RNA? Because RNA was already measured and is often measured and in the forefront of many people's mind. What about RNA? I do not believe RNA is a great surrogate for protein, so I always look at both, because transcriptional regulation is different than fundamental signaling regulation or proteolysis, mediated decay. Those types of protein level regulation events, they're different. And so the abundance measurements that you get for protein and RNA will be different. And what I can see that I'd like to show you is here in the RNA graph, chromosome 8 really pops out as having a strong line. And the protein graph, that's not there. It's much weaker. So that was predominantly transcriptional regulation activity. It's great for us to be able to characterize locuses that are acting as a transcriptional regulator versus locuses that are acting as a translation, as a protein level regulator. So I've blown up a little bit of this. This is just chromosome 2. And in reddish, I plotted the trace for mRNAs that are affected. So this is every single locus, how many other mRNAs it affects. And in blue, I've overlaid the proteins for the same locus. You have some places like this end where the mRNA and the protein track very same. These are mostly cis regulation here. There's nothing going on in terms of trans that's exciting. You have other places where they're very different. And the granularity that was given to us is not superb, but you see a spike, and then it drops down, and then another spike of regulation, and it kind of drops down, and then another big spike of regulation. So there is some ability to go back and look at the data and say, what are these proteins and why is SMEC2 affecting 300 other proteins? What are the proteins it is affecting? Why are they contributing to the phenotype of ovarian cancer? It's just another example. I show this one mostly because people ask about BRCA1. BRCA1 is important in lots of cancers, including ovarian. People want to see that. There was nothing that stood out to us about the number of proteins or any functional enrichment as of yet with BRCA1, but we continue to look at it. So here's how we do functional enrichment. Oh, sorry, go ahead.
So how homogeneous are the samples that generate this data?
What do you mean homogeneous?
Or I guess heterogeneous in terms of the actual samples that are sequenced. So are all these patients at exactly the same stage and they're exactly the same age and they're exactly the same, and sort of and how do those variations,
sure,
sort of where people are in their disease course affecting?
So the ovarian cancer cohort of TCGA is about 500 women, and they are selected for high-grade serous ovarian cancer, which is one subtype of ovarian cancer. For those of you who are familiar with breast cancer, there's multiple clinical, luminal, basal subtypes, and all of those are sampled by TCGA. For ovarian, they went one focus, and they just did high-grade serous ovarian stage 2 through 4. That is a pathological. These are mostly older women, though not all. They are not selected for uniformity. Candidly, TCGA was selected for convenience because the NIH had a ton of money it had to use because of the stimulus. So they said, let's get out to every hospital in the country and get cancer. Let's pull tumors out of people and characterize them. These have all gone through some histology quality checks in terms of tumor nuclei count or heterogeneity or necrosis. And so the TCGA set has been checked for that, but it is not homogeneous by any stretch.
Results would be markedly different if, do you think there's a lot of heterogeneity in there, and that, a lot of the signals that you're getting, especially like BRCA1, for example, would it be better if you could have some subsets of groups that were more clinically meaningful?
So in speculation, I would say it would always be better to have more homogeneity. One of the things I can say is that the data that we see here is as good as it is because of the power that we have from 174 tumors. So the breast cancer sampling, for example, is fewer. They only have about 100 or 80 tumors. The ovarian cancer had twice as many because we had two centers working on it. We had the Hopkins and the PNNL Center both working on it, so we combined efforts and sampled more tumors. And if I downsample my data to just the PNNL or just the Johns Hopkins data, you see far fewer statistically significant correlations, so it all gets kind of dropped to the floor. I wish we had 500 tumors. Also, the one thing that I should answer in terms of your question is PNNL selected tumors based on survival. So we tried to pick the very worst, shortest survival and the very longest survival. So we stratified that. And Johns Hopkins picked tumors based on their marker for chromosomal instability, which is BRCA1. So anybody who had a BRCA1 mutation, they picked them. And then we randomly sampled to fill in the rest. Were there other questions about the data? Yeah.
So how do you quantify protein variation?
How do we quantify protein variation?
Yeah.
So this data, what we did is you get an abundance value, which is measured on, if you are familiar with mass spectrometry data, it's the intensity of the ions that come out for all peptides and all tumors. And then that is normalized through some rather exotic and complicated statistics where we normalized both within our center, then we normalized with Johns Hopkins to be across the center to make sure there wasn't a substantial batch effect. It took like 3 or 4 months actually to do that process, so something we thought a lot about. It's not, I actually have a lot of people ask me, how come you can't just give me read count? Because they're familiar with RNA-seq and they're like, just give me read counts, it's so easy. I'm like, well, we're a different type of data, it's a lot more descriptive and it's a lot more difficult. So there's a heavy amount of statistics for those of you who are statistically oriented that go into this. Any other questions before I move on? I love questions, by the way. My lab at UCSD was you prove your worth, right? So there was interrupt driven. I have no problem with that.
Heterogeneity, when you look at the coffee number,
Yeah.
variations
Yes.
across in this cohort. So how much? I mean, let's say if you evaluate, certain region, its frequency, among 100 or 200 patients. So what is that like? Like the copy distributions,
So these are real values. Unfortunately for my display, the data by TCGA has been log transformed and normalized, so it's a variance stabilizing transformation. But
the average of the change across
this is one person and their value for the TGIF locus, normalized against every other tumor in the cohort. So haplonormal, what I would consider diploid-ish, would be this. These are people who have 2, 3, 4, 5 copies. These are people who have partial copies or 0 copies. It's not a specific number because they're converting the read depth from RNA-seq reference mapping into a number. And then they're transforming that number to give it some, centered around 0 mostly.
The following heat map, is that on the average effect or it's on the...
So this is what the heat map is showing. This person right here, this combination of locus and protein abundance has a correlation that is statistically significant and positive. So in the following heat map, the XY coordinates for this locus and that protein will show up red. This has a statistically significant, because we do the Benjamini-Hochberg multiple hypothesis testing and all that, but this is negative, so it will show up on the next plot as blue. Because it's a negative correlation. So what you're seeing here is not an abundance value, but simply a positive or negative correlation that passes. So this locus has an effect on this protein, and it's positive. So it shows up in red. It has a blue negative effect on this protein. So that's how that's built up, which is why in the end we just sum down and we say this locus affects 300 other proteins. That's awesome. This locus affects really only 20 or 30 other proteins, so we're not going to spend our attention on it. These are great questions because it's the crux of what I was getting at, so Anything else on this before I move on? Yeah, go ahead.
It might be a basic question, but how could the copy number be non-integer?
So copy number is non-integer because chromosomal abnormalities are not done by students with PCR primers. Right? When you're PCRing something, you put in 6 copies because you built it. Right? Tumors have problems in their polymerases, they have problems in their end joining, non-homologous end joining, and stuff happens. And so you get 2 exons of a protein repeated 15 times and none of the rest of it. So the average read count for that gene is above 2, above diploid. It's not 3 and it's not 15 because you only have a couple exons, not the whole thing. It's not a clean copy. That's the, and when you get down and you look in the data, this is just what it looks like. Anything else? All right. So functional enrichment, what we do is we take the proteins which have been affected in trans, and we just do a Fisher's exact test to say, are these proteins enriched for any set of proteins who someone has told me this is a set? So we can use GO if you like GO. We can use KEG if you like KEG. We can use the NCI pathway database. We can use Reactome. Wherever you get a gene classification or a gene grouping, we can just test that. We can say, I found 350 genes in this enriched in this trans set. Are they enriched in any function? That's pretty easy. The hardest part is actually finding a good classification because that's the grunt work that people have to do and don't do since 1970. So what I can say is that we see, I would say, thematic enrichment. So since KEG isn't perfect and KEG isn't built for tumor pathways, it doesn't obviously pop out and say, this is aberrant, this protein and all of its downstream transcriptional targets. Nobody's gone through the curation of that. But here what I see is migration and phagocytosis and cytoskeleton. So in that group of proteins, this reminds me of invasion and metastasis. Those are proteins which would be nominally relevant to that as a process. Similarly, on other locuses, I find things that look like immunity-driven genes. So KEG will show it up as asthma and B-cell receptors and IG production. And all of these are big pathways in KEG that have hundreds of genes, but they seem immunity-related. And this is where my background in cell biology stops. And I have to start working with collaborators who are cancer biologists. And I have to say, help us interpret this data in a meaningful way because I have reached my pay grade and I'm able to show you that this locus has significant effects and it seems thematic for me, but I need you as a clinical oncologist to push it the rest of the way. And that's a handoff that we're doing right now with the people in our ovarian cancer group. And that handoff has driven me to the next set of slides because it's very difficult because I just spent 30, 40 minutes explaining what I did to you. I typically, when I do this with our collaborators, I spend an hour multiple times over the phone, or in person. And it just takes a long time to describe what we're doing, why we're doing it, and what we think the data means. But the thing that I wish in my Christmas wish list is that there was a classification that was better than KEG. But in all honesty, I think KEG's as good as it is right now for most things. So the last thing that I want to say is we find things that are not the usual suspects. Here on chromosome 12, we have KRAS, FOXM1. These are things that people knee-jerk think about in cancer, and they show no effect, either at the mRNA or at the protein. There's nothing doing. Changes in their copy number doesn't really drive problems elsewhere in cell. And that was surprising to us. We found proteins that we have no idea what they're called because they're not called anything real, right? This is just the name of a cDNA somewhere. And this protein's never been studied, but it affects 190 things. So there's a lot of new biology that we believe this uncovers, and we believe it points to new areas for people to spend time thinking about what is happening here. And so the last little part of my talk, I'd like to talk about solutions for large-scale collaboration. So CPTAC as a cancer biology project is one of many large-scale projects that we work on at PNNL. We are, as I said, a team science group, and we are intensely collaborative. We currently have a large project. The first time I talked to someone about TEDdy, they said, my son's in that. So I don't know if anyone here, their child has type 1 diabetes and is under the age of 10, but this is a project that studies environmental determinants of type 1 diabetes. And so they enroll people who are genetically predisposed or who have siblings who have, diabetes, so that they can track them from birth till onset. And they have very good long-term monitoring of these patients. It's wonderful. It's a huge data set. PNNL will be involved in creating all the proteomics data for them. This involves blood samples every 6 months from birth to 15, if that's when they develop the disease. And they will also have transcriptomics and genomics and metabolomics. They'll have all of this and their clinical data. And it's a huge project. When I asked once who would I talk to about TEDdy, they were like, well, there's hundreds of people. I don't know which one you want. I'm like, well, I want to talk to the one in charge. And they're like, well, it's like 6 of them. And so it's a big thing, but that's one of our great collaborations. We also have a project with the University of Miami. It's an Alzheimer's project that studies aging men, both those who have Alzheimer's, but those who have other dementia-related diseases and those who don't, and some monks in Europe. So they're great controls, and all of these projects that are on this page have genome, transcriptome, proteome for hundreds of people. And this is the amount of data that typically starts causing problems. In many of these projects, I anticipate all of them and all the others, which I will deal with in the next few years. I will get a phone call at some moment that says, can you do it? You gave us the data. It's too much. Can you do it? I take some of these phone calls personally. I take some of them through the other people who work computationally. Some of them Dick Smith takes. And, it's just they call us and they say, you transferred us the data, and it's a spreadsheet of 10,000 columns and 400 rows, and you told me that was the good stuff. I don't know what to do with it. And I gave it to my computational people, and they only know about RNA-seq. They don't know what to do with it. Can you do it? Now, I am not able to solve those diseases because I don't have a 20 or 30-year background in that disease. Right? I'm not going to cure ovarian cancer, as I just said. I'm confident that my collaborator at Sloan Kettering, he's going to do an awesome job with it. And I want him to look at our data in a meaningful way. It's that very overused phrase, what is a meaningful way? So this has been the focus of the obsession of my last 6 months. I think there is more that we can do than simply pushing data through our pipe and making servers. Right? That is where I think the shoe drops and people say, you got to do more for me. And it's paradoxically not more computer science. It is presenting the data in a better way so that somebody who has a 30-year background in diabetes can look at it and use it and then discover. Right? They're going to do discovery on their own time scale, and it's not going to be because I handed them a terabyte. That actually makes them pretty mad. But this is the little graphic I did for the poster, and as I said, this is something I hear often. I am the guy in KEDS and a Mac. Hey, you're the disease specialist. You guide me, tell me what's important, and they just say, tell me what's significant. And multiple phone calls, like months of phone calls go on in this. So data exploration is currently terrible. And that's because data exploration is searching through Leninger's biochemistry, Googling, throwing up chart foo in R, or looking at something in Excel. And all of these are bad because none of these are the right way to look at data. Right? They all compartmentalize the wrong pieces, the wrong places. Sometimes we show heat maps. I showed you a heat map. I'm mea culpa. I'm part of this. But this heat map is static. And so one of my many aha moments was when somebody in our group meeting said, "What's this one?" And it was a TIFF, right? It's a picture. So we go back through Excel and we retrace and we try and find this one. Answering that question, what's this one? Takes a long time. Or who's this patient? It looks like she doesn't quite belong. I would have put her over here. Right? Answering that question takes a lot of effort because you're dealing with some R, some Excel, some graphics, static graphics. And that's never going to solve the problem. So I have spent 6 months and I am in the planning stages. So this is by no means finished. I have not solved or climbed Mount Sinai. I am not ready yet, but I wanted to talk to you because I want to say this is where the future of bioinformatics lies. When I entered graduate school, there was microarray and PDB, and we were chunking and pushing data, right? But the future of bioinformatics is helping find discoveries. I feel very strongly about that we need to change the balance of who's in charge. So what I want, and I've started building just a simple kind of PHP interactive graphics, I want somebody to be able to click and say, these patients, they separate out. Who are they? It says, oh, that group of patients, here's the data if you want to pin it to a canvas over there. You know, I'm Pinterest, me and my wife, we love it. But the distinguishing metadata on these people is that they survive. They're alive for 4 years after the disease. This is our good cohort. In all honesty, when you look at the static heat map and you say, who's the good side? Right? You can't answer that basic question without going back to an Excel spreadsheet, tracing it to a different Excel spreadsheet that has the metadata. But I want to just fastly click. And I want to let my cancer oncologist, my research scientist at a med school, click. Who are these people? I want that kind of access. I want the kind of access that says this row of proteins that appears to be up in my good health outcome people. What is it? I just want that to happen. I want it to happen at the speed of clicking. I don't want to have to pre-populate databases, or I don't want to have to call somebody on the phone. I want it to just happen. It has to happen at their curiosity. Their curiosity is going to be sometimes very fast, sometimes very pensive and slow, but I want them to be able to do it, not calling me. I want it to show up on a map that is contextually and visually obvious to them. So if it's metabolism and it's the TCA cycle that shows up and I have both metabolite and protein data, I want it to show up. Say, this is what I found in these patients who have good outcome. 2/3 of the way to achieving this already, but this is only the start. This is the genesis of what can you do to give someone data in a meaningful way. When I left graduate school, Steve Briggs, who is my advisor and who did the rice genome, said, I don't know what data visualization is, but I know it's not spreadsheets. If you're giving me a spreadsheet, it is a wrong choice. So I have been thinking about this for many years. And it has finally come to a head when so many people just said, do it for me. And I realized I'm not that person. At first, it's really flattering. You know, somebody say, you're going to solve this disease. And you're like, no. No, you are. Because you're the one who spent time on it. Anyway, that's where I'll stop. But I have lots of ideas. And I know I'm speaking with some of you at lunch today or later today. But I want data analysis to be a focus, not data processing. And the NIH has started to make changes in that. They recently have the Big Data to Knowledge, right, as a trans-NIH project. They're starting to realize that they've got lots of data and the knowledge it's generating is low. You know, the big PSI structural biology grants, when they're renewed now, they say, we're not going to be making structures so much as we're going to be helping you learn to use them. Right? There's a PLOS biology paper where they're like, we made so many structures and none of them are published. Right? They're making data, but it's not translating into useful knowledge. And I think it's because data analysis has not been a focus of people's effort. So I am now part of that team that is moving towards data analysis, computationally assisting. So Debjit and Junyang are my postdocs. Debjit worked on the ovarian cancer story. Junyang has, in very short order, put many of my ideas about interactive and intuitive exploration of data into practice. Nick Kramer and Alex Ender are actually PNL's group of visual analytics. So we have probably 50 or 100 people whose job it is to make visual analytics tools, and they do it for a variety of national security related solutions, but it's the same thing. You need someone who only understands Uzbekistan and doesn't care about what a support vector machine is. They just want to have data come and be useful. Dick Smith and Karen Rodland are my bosses in the sense that they would be like a chair of a department for all of you. They're great and they're supportive. This is my lovely family. My wife and 4 kids. We love it in Washington. And I have postdocs and summer interns that I would love to host. So if any of you feel intrigued by any of this, any part of it, I would love to be part of your scientific future. And thanks. Thank you.
Any more questions for Sam?
Yeah.
I work in a translational lab.
Uh-huh.
I do background in computer science. And right now, for the last 4 years, I work actively with doctors and biologists. And the problem is there, clearly. But I do think the solution is in data visualization. Because even with the best software, with the best visualization, with best everything that you provide to these people, they don't feel comfortable. And many times they make wrong interpretations.
Yes.
Because they don't know the strengths and weaknesses of the data.
Yes.
Because the data we generate is not perfect data. There are lots of limitations of it. And we are aware of it because we generate it and we use it.
Yes.
But they don't. So I think, if you ask me, the solution is somewhere creating positions for people who can bridge between these two sides and provide actionable information to people who will apply these solutions to patients.
Yes. And so in my project with the Visual Analytics Group, which is a national security project, we focus quite a lot on doing high-powered calculations that support people. A lot of that is meant to be transparent and intuitive. And one of the things that I'm studying actually right now is identifying bias. So when someone is visually analyzing data and they're hunting down a tree that's obviously biased, how can we alert them that what they are doing is overly biased? They're not making full use of the data, they're misunderstanding the caveats of data analysis processes or data processing steps. Right? So that's one of the things that I think is very important. I agree with you that we can't just say, tell me if P53 is important, and they'll say yes. Right? You have to provide the background, and that's why visual analytics is a science. And interaction is a science and not simply web programmers. Right? If you're just providing a GUI, you might as well be providing an active version of Leninger. Right? Flip to Leninger page 5, do it. Right? But we want to provide the underlying statistical confidence for their exploration. So, go ahead.
Another question. So there is limitation with any kind of data, including proteomics. And one particular limitation is there are so many alterations on those proteins.
Oh yeah.
Phosphorylation and other things. So it's difficult to decide which measurement is more important.
That's true.
Functional role. Are you studying that problem as well?
So with our data, we have done both a global proteomics, which will give you simple protein measurement abundance, and we're doing a phosphoproteomics, which will give you phosphorylation stoichiometry. Stoichiometry as close as we can get it. I mean, we didn't actually count with eyes and little gnomes, but as best we can do, we normalize phosphopeptide abundance to the global protein abundance, so we can say this protein at this site is more phosphorylated than simply rising protein abundance. It's stoichiometrically more phosphorylated. So the guy who works with me on that, who's not part of this presentation, says phosphoproteomics is the winner. It is the most information rich. It provides the best explanation, in our case, for survival in ovarian disease. So our hope is that we can, and this is why my boss is building new tools, new toys, we can measure all of those at the same time. Right? So when you run an Illumina machine, you have to run it differently for methylation data than you do for RNA data. You do your sample prep a little differently, you run the machine a little differently. And his goal as an experimental instrument developer is to make an instrument that can measure all of your data at the same time. It can measure metabolites, proteins, phosphorylations, all of it. And that's one of the reasons I moved there, to be honest, because he's making new machines that will hopefully at least try to address this question. Any other questions? Yeah.
So, yeah, I really, I feel your problem here, of how you enable, clinicians who are, have the domain knowledge to be able to explore the things that they have. And I think, so I teach a course here where we try to sort of go in a different direction. And I think part of the problem in my mind is a generational issue. Because, the folks that are really experts right now are, 30, maybe 20, 30 years into their career. And, at some point in time, they had to learn methods in biology so that they could do their wet lab stuff.
Yeah.
Right? And so I think that we're at the beginning of a point where, data analytics and data analysis is going to be part of, teaching curricula for, future MD masters or MD, researchers. And that I'm hoping this will help, close that gap. And so what we try to do here is, we actually have a course where, I have fellows that are starting the research year, and we're teaching them data analysis. We get their feet wet, and we just throw them in there and give them a big data set and say go, and they start exploring. And I'm not quite sure how much, from a theoretical perspective, how much of that can be offloaded, to things, where you can just say, oh, run logistic regression, and then it just does it, versus really always needing the person who's, really interested in the problem and actually doing the problem.
Yeah. It's very tough, and we run into a lot of those. So in our ovarian cancer data, as an anecdote, we found 0 new subtypes, and our program officer was not happy with that. Many times on, I would say, every biweekly phone call, he's like, what new subtype did you find? Because he saw in colon cancer that was a great thing for people to understand, and so he wanted to know if we found it. And we looked and looked and looked, and sometimes we would think maybe this might be justifiable. And then, different statistics analysis would say, no, not so justifiable. So there is a moment where you do need someone who is classically trained to really get into the depths of that. But I do want people to be aware of data, and I think that is happening. And, I think people who graduate are being skilled in at least understanding the basics of algorithms so that they can not, cut their own throat.
Well, I think things will stand the test of time. I think there will be techniques in physicians' toolkits. So like logistic regression is, a technique that's just accepted, it's been through its paces in the last 30 or 40 years. And I don't think MDs are necessarily going to be, unless they're doing it for their research, doing the cutting edge stuff. Because there's too many unknowns about the stability of those algorithms and, when they work and how they work under what conditions, where you can just assume, accept anything this thing says. But I think over time, there are going to be techniques that are going to be, pretty rock solid. And those are the ones that are going to filter down, to the physician. You just say, just use this.
Yeah. I'm hoping to be part of those on all sides, like both presenting it to a physician and also working on the underlying statistics. My story that I tell a lot is BLAST. BLAST revolutionized sequence alignment. It was great. It was a big leap over the Smith-Waterman alignment. Not in alignment, but in confidence. Go ahead.
When making this kind of pipeline, do you think it is critical to make it incremental? Whenever new data comes in, I'm ready to take it and updating my whole model and give new advices.
So that's exciting because that's the project I also work on with the National Security Division at PNNL, is analysis in motion, streaming data. So streaming analytics. I think for some applications that's going to be real, and that's going to be very important. For some planned biology experiments, that may be more or less useful because we have a cohort of 174 people. And we did some preliminary analysis when we got 50 of them done, but we waited until the end of the experiment to really write our paper, right? And so I think streaming analysis will make sense for some contexts and not others. That's as best as I can do, although that's something that I play around with for maybe 5 or 10 hours a week. Go ahead.
You said that in the results of your analysis, you had some new potential candidates.
Yes.
As seems for the first one in Omics. But you didn't get some of the usual suspects.
Yes.
So how do you explain that? Because I mean, in molecular biology, people that so clearly some of the things play a role. So how do you explain that you don't see that?
So on that specific one, it's the type of analysis we're doing. So KRAS, in that example, does not affect global protein abundance. It might affect phosphopeptide or phosphorylation abundance. That's not in this analysis chart there, and we're working that up. But for whether it changes the abundance of lots of other things in the cell, that's clearly no. And it's just a different view. And I looked at this big picture I have here. So this exists for the colon cancer paper. Whoops. And it looks very different for colon cancer. We actually asked my postdoc to overlay the colon cancer findings and our findings in a N of 2 pan-cancer paper. And there is very little overlap. And so whatever dysfunction is happening in colon cancer is not happening in our ovarian cancers. And I wish I knew more about that, but I'm not a cell biologist, and so we work to explain those things as best we can. Anybody else? Well, I really appreciate your time.